Hello friends, now we are going to discuss citric acid cycle, TCA cycle, Krebs cycle. Three names. Citric acid cycle. Other name is tricarboxylic acid cycle. Tricarboxylic acid. So you can understand from this point that citric acid is tricarboxylic acid, means there are three carboxylic group in citric acid. So this is TCA cycle. Another name is Krebs cycle. We will make mnemonic that TCA cycle T4 TCA T4 10 ATP. It produced 10 ATP from one acetyl coenzyme. So it is the final pathway for the metabolism of carbohydrate. We have discussed it in a sequence. I told you glucose is converted to pyruvate by glycolysis, aerobic glycolysis, 7-AT produced, pyruvate enter mitochondria. In mitochondria, pyruvate is converted to acetyl coenzyme by pyruvate dehydrogenase. Pyruvate dehydrogenase link reaction. Now acetyl coenzyme is produced in mitochondria from pyruvate. Now this acetyl coenzyme take part in TCA cycle. So I can simply say that glucose or carbohydrate final oxidation cycle is this TCA cycle, carbohydrate. So I have discussed it with continuation of carbohydrate. Doesn't mean that it is only final oxidation pathway of carbohydrate. It is also oxidative pathway, final oxidative pathway for lipid and protein. In lipid, you will find beta oxidation of fatty acid produce acetyl coenzyme, and this beta oxidation occur in mitochondria. So this also occur in mitochondria. I have written over here, but beta oxidation also occur in mitochondria and acetyl coenzyme may produce. This acetyl coenzyme may take part in TCA cycle. Proteins are made up of amino acid, ketogenic amino acid, leucine and lysine. Lily, don't be silly, mnemonic. They produce ketone bodies. Ketone body produce acetyl coenzyme, and so acetyl coenzyme enter Krebs cycle. So you can say glucose, lipid, protein, all these final oxidation cycle is Krebs cycle, TCA cycle. We found that all the enzyme which we are going to discuss in Krebs cycle, they are present in mitochondria. All are present in mitochondria. And we found that all of these enzymes are present in mitochondrial matrix except all enzymes of Krebs cycle present in mitochondrial matrix except succinate dehydrogenase. Mnemonic. It is sucked in. Succinate, mnemonic, sucked in inner mitochondrial membrane. Inner mitochondrial membrane. So succinate dehydrogenase is present at inner mitochondrial membrane. All other enzymes of citric acid cycle are present in mitochondrial matrix. So this is mitochondria, and now we start citric acid cycle. Starting point is oxaloacetate, first substrate for Krebs cycle. If I say first substrate for Krebs cycle is oxaloacetate, although first reaction is oxaloacetate bind with acetyl coenzyme to form citrate or citrate. Okay, so you can say acetyl coenzyme and oxaloacetate both are joining together. So both are first substrate, but one to choose, I will choose oxaloacetate. One reason is that in most of the textbook, it is written that first substrate for Krebs cycle are oxaloacetate plus acetyl coenzyme. One reason I am giving. Yes, gradually you will understand why I said it the first substrate. Okay. So question has been asked long time back. If question is asked first substrate, I will prefer to mark answer as oxaloacetate. Clear? Now, Acetyl coenzyme may bind with oxaloacetate. Acetyl coenzyme is having 2 carbon. We have already discussed. Pyruvate 3 carbon is converted to 2 carbon acetyl coenzyme in link reaction. Oxaloacetate is having 4 carbon. Many times I write X like this. 4 arms, 1, 2, 3, 4. So you will remember number of carbon and this Krebs cycle would be very simple. Oxaloacetate is having 4 carbon, aspartate is having 4 carbon. So no need to worry. We will discuss all these metabolisms for the time being. Aspartate is normally converted to oxaloacetate. The only difference is ammonia. So aspartate is having 4 carbon, oxaloacetate also having 4 carbon. Just to introduce the number of carbon of oxaloacetate, 4 carbon. 4 plus 2, citrate definitely must be containing how many carbon? 6 carbon. By citrate synthase, this is irreversible step, irreversible, and this is regulatory step, regulatory step of Krebs cycle. Now citrate is converted to isocitrate by enzyme aconitase. Here also I will write aconitase. Same enzyme catalyze 2 steps. Same enzyme catalyze 2 steps, so don't get confused. If you see in many questions, they will give that citrate is converted to isocitrate. They will omit this intermediate step. And you will see citrate to isocitrate. Citrate and isocitrate are isomers. Definitely it is having 6 carbon. So enzyme should be isomerase. No, in these two reaction, this aconitase is a lyase. I write not isomerase, not isomerase. Question. How you will remember that citrate is converted to cis aconitate by enzyme aconitase, cis aconitate. This cis aconitate is converted to isocitrate by same enzyme aconitase. You will remember mnemonic a crow was thirsty. He was dehydrated. Dehydration. So first reaction is dehydration. Now he took pellets of iron. In place of stone, he took pellets of iron. So why I said iron? Because yes, aconitase is an enzyme which contain iron. Now this thirsty crow drove this iron and now pani comes and now he become hydrated. So this next step is rehydration. So you will never forget about aconitase now. This aconitase is inhibited by fluoroacetate. This question is asked. Now, this isocitrate having 6 carbon is converted to 5 carbon compound alpha ketoglutarate. You will remember alpha ketoglutarate, glutamate, glutamine, all are having 5 carbon. Again, I am repeating glutamine, glutamate, alpha ketoglutarate, 5 carbon atoms are there. So a 6 carbon compound isocitrate is converted to 5 carbon compound alpha ketoglutarate. So 1 carbon must be removed. Removal of carbon or removal of carbon dioxide is called decarboxylation. So when isocitrate is converted to alpha ketoglutarate, in this reaction CO2 is removed and this step you can say there is decarboxylation. Correct. What is the name of this enzyme? The name of this enzyme is isocitrate dehydrogenase. And I have told you wherever dehydrogenase is there, dehydrogenase means removal of hydrogen. Hydrogen will be taken up either by NAD, FAD or NADP, most commonly NAD, to form NADH. NADH. And you know, removal of hydrogen is called oxidation. Joining of hydrogen, reduction. Removal of hydrogen, oxidation. So in this, hydrogen is removed and removal of hydrogen is oxidation. So you can say when isocitrate is converted to this isocitrate is converted to alpha ketoglutarate, there is oxidation, there is decarboxylation. So you can say this reaction catalyzing oxidative decarboxylation. Oxidative decarboxylation. A frequently asked question that oxidative decarboxylation cofactor is vitamin B1. I have discussed it. Yes. Remember link reaction? I told you. No, 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 no. In this, B1 is not cofactor. You know that complete statement is B1 is cofactor for oxidative decarboxylation of alpha keto acid. This isocitrate is not alpha keto acid, not alpha keto acid. So B1 is not cofactor for this reaction. This is very, very important question. So what are the cofactor for this reaction? The cofactor for this reaction are Mn, Mg. Clear? So question is asked. Now, alpha ketoglutarate, how many carbon atoms are there? 5. This alpha ketoglutarate is converted to succinyl coenzyme A, and if I write U like this, you will say it looks like 4 carbon. Succinate is having 4 carbon. So in this reaction, alpha ketoglutarate is converted to succinyl coenzyme A. A 5 carbon compound is converted to 4 carbon compound. So 1 carbon must be removed. And removal of carbon dioxide is called decarboxylation. And you know, the name of this enzyme is alpha ketoglutarate dehydrogenase. Dehydrogenase. Yes, alpha ketoglutarate means substrate. Okay, many times in enzyme you will find the name of substrate alpha ketoglutarate and the action of enzyme dehydrogenase. So it remove hydrogen from alpha ketoglutarate. So you know whenever there is removal of hydrogen, there is oxidation. So you can say in this reaction there is oxidation. So you can say this is oxidative decarboxylation. Yes, oxidative decarboxylation of alpha keto acid. Yes, alpha ketoglutarate name itself tells you that it is alpha keto acid. So, and whenever there is oxidative decarboxylation of alpha keto acid, cofactor is vitamin B1. So, in both of these reaction, yes, both of these dehydrogenase, CO2 is removed. In both of these dehydrogenase, oxidative decarboxylation occur. But in isocitrate dehydrogenase, cofactor is not B1, but in alpha ketoglutarate dehydrogenase, cofactor is B1. Question is asked. One question is asked in Krebs cycle or TCA cycle or citric acid cycle, one CO2 is removed just before and after alpha ketoglutarate. Now you can say CO2 is removed, definitely removed just before and after alpha ketoglutarate. Yes, this question is asked. So till now, I have removed all the carbon. Yes, acetyl coenzyme A. How many carbon were there? 2 carbon. So I removed these carbon in the form of CO2, one CO2, second CO2. Now 4 carbon succinyl coenzyme A is produced. So till now, I can draw a line. Till now, yes, whatever oxidation, whatever carbon removal was about to done was done. Now the rest of reaction The purpose of rest of reaction is to produce oxaloacetate, means to regenerate oxaloacetate. Yes, succinyl coenzyme A 4 carbon, oxaloacetate 4 carbon. So all the intermediate are having 4 carbon, means in all these reaction there would be no change in the carbon. There would be change of hydrogen oxygen, but no change on the carbon number. So I can say succinate is having 4 carbon, fumarate is having 4 carbon, malate is having 4 carbon, oxaloacetate is having 4 carbon. So again repeating, malate is having 4 carbon, malate is having 4 carbon, malate is having 4 carbon. What are you doing, sir? Yes, I am making you aware of a very important question. Okay. Anyways, you will appreciate soon why I am repeating this again and again. Yes. Now, I will write few questions of these two reaction. Isocitrate dehydrogenase, this is also regulatory step. This is also regulatory step or regulatory enzyme. This alpha ketoglutarate dehydrogenase is also regulatory enzyme. Regulatory enzyme. So till now, I have discussed three regulatory enzyme of Krebs cycle. One was citrate synthase, regulatory step, regulatory enzyme. Second is isocitrate dehydrogenase, and third is alpha ketoglutarate dehydrogenase. I told you that this citrate synthase catalyze irreversible reaction, and I will mention here that alpha ketoglutarate dehydrogenase catalyze physiologically irreversible reaction. Question. Now we come to succinyl coenzyme A to succinate. Succinyl coenzyme A to succinate, and the name of enzyme is thiokinase. I have discussed multiple times, all kinases are transferase. All kinases require magnesium as cofactor. So thiokinase is transferase. Thiokinase require magnesium as cofactor. Whenever kinase is there, either ATP is used or produced. In this, ATP is produced. And you know, if kinase produce ATP, that is called substrate level phosphorylation. I have already told you the difference between substrate level phosphorylation and oxidative phosphorylation. Again, if any reaction occur and energy is released, this energy is directly converted to ATP at the level of substrate. Just like here, that succinyl coenzyme A to succinate, energy is released that is directly used for the conversion of ADP to ATP. That is called substrate level phosphorylation. If I talk about the previous step, in this reaction, alpha ketoglutarate to succinyl coenzyme A, yes, energy is released. That energy is conserved in the form of NADH. And you know, this NADH will go in electron transport chain to produce 2.5 ATP. In electron transport chain, there would be oxidation and ATP will be produced, matlab phosphorylation. So that is oxidative phosphorylation. Got the point? So oxidative phosphorylation, electron transport chain is used. In substrate level phosphorylation, energy is released. At the same time, it is converted to ATP. Thiokinase, in this, if you see this, I have taken both GTP and ATP. Most of time in standard textbook, you will find they have taken only GTP. But you will remember thiokinase has two isoenzymatic form. One produce GTP, second produce ATP. Or you can say thiokinase can produce GTP or ATP both. I will try to integrate. Gluconeogenesis, we will discuss synthesis of new glucose. In synthesis of new glucose, we require energy, we require GTP. You know, gluconeogenesis occur in liver and kidney. Yes, just mug up. If you don't remember, it's okay. For the time being, just mug up. Gluconeogenesis occur in liver and kidney, and gluconeogenesis occur in starvation. So in starvation, there is no glucose. Yes, you are not taking diet. So liver will try to maintain blood glucose by synthesizing new glucose. Kidney also help liver. Liver is the boyfriend and kidney is the girlfriend. So majority gluconeogenesis is done by liver. A little bit work is also done by kidney, the girlfriend. And this occur in starvation. They contribute blood glucose by gluconeogenesis, and gluconeogenesis one step requires GTP. So during starvation in liver and kidney, this thiokinase will produce GTP. Normally it produces ATP. So this GTP production is a specific feature. That's why in books they have mentioned this GTP. So don't get confused. Questions are asked. You will solve question according to the concept. Now, succinate is converted to fumarate by succinate dehydrogenase. Now there will be no change in the carbon because succinate is having 4 carbon, fumarate is having 4 carbon. So no change in the carbon. Only hydrogen will be removed. Dehydrogenase, removal of hydrogen. Hydrogen will be taken by FAD to form FADH. Mnemonic, most of time you will find that dehydrogenase is there, hydrogen is taken by NAD to form NADH, but succinate, suck, fuck, yes, produce FADH. छी गंदी बात. Okay, yes. So fumarate is produced and FADH is produced. And this succinate dehydrogenase is inhibited by malonate. Yes, if I ask you, think in your mind. Yes, malonate is having how many carbon? Malonate is having how many carbon? Yes, 3 carbon. Malonate and malate are different. Malate is having 4 carbon. Now you understand why I repeated multiple time. Yes, malate is having 4 carbon. Melon, watermelon, having how many carbon? 3 carbon. Yes. So question is asked. Succinate dehydrogenase is inhibited by option A, malate. Option D, malonate. We are so preoccupied with M. As we see M, we mark malate. No. Read the question complete. Yes, and you will find malonate in the option in the last. They know that you may commit mistake. So malonate is having 3 carbon, and I told you it inhibits succinate dehydrogenase. Other than succinate dehydrogenase, it also inhibit complex second of electron transport chain. Third, it also inhibits CPT1. What is CPT1? We will discuss in beta oxidation of fatty acid. So CPT1, succinate dehydrogenase, and complex 2. In fact, succinate dehydrogenase is part of complex 2, and I told you succinate dehydrogenase is sucked in inside the inner mitochondrial membrane. So it is the only enzyme which is present at inner mitochondrial membrane. Question. All other are present in mitochondrial matrix. This fumarate is converted to malate. This malate is converted to oxaloacetate by malate dehydrogenase, and you know that wherever there is dehydrogenase, hydrogen is removed. Removal of hydrogen is called oxidation. Oxidation occur. So in this, hydrogen is removed. Hydrogen is taken up by NAD to form NADH, and oxaloacetate is produced. So starting point was oxaloacetate and the ending point is also oxaloacetate. So oxaloacetate is regenerated. You know, if any reaction is occurring and you put catalyst, catalyst is not used in the reaction, in the same way it will appear that oxaloacetate is not being used. It started with oxaloacetate, ultimately the same amount of oxaloacetate produced, so it act as a catalyst in Krebs cycle. So oxaloacetate act as a catalyst in Krebs cycle. Question. Now I will give little bit more importance to this reaction. This is reversible reaction. What is specific in this reaction? We will use it in malate et al. So you must have an idea. When I will discuss malate shuttle, at that time it would be easier. So normally malate is converted to oxaloacetate. When malate is converted to oxaloacetate, reaction is going in this direction. NAD is converted to NADH. NAD is converted to NADH. But this is reversible reaction. If this reaction occur in this direction, then oxaloacetate will be converted to malate and NADH will be converted to NAD. Just have this information in your mind. We will use it again. Not so important for direct question, but important for malate shuttle. Now we solve this question. A 48-year-old male with excessive drinking means alcohol, water is not considered as drinking, okay, over an extended period of time while eating poorly. This is myth that if you take alcohol and take a good diet, alcohol would be harmless. No, alcohol is harmful. Okay, the only benefit of taking good diet with alcohol is that you will get nutrition, particularly vitamin B1, thiamine, because alcohol cause B1 deficiency. Very important. So if you take good diet, so thiamine deficiency can be delayed. Okay, not prevented at all. So anyways, so a patient is taking alcohol for a long time, now admitted to the hospital with high output cardiac failure. And you know, whenever you will see high output cardiac failure and thiamine deficiency, B1 deficiency leading to wet beriberi. So which of the following enzyme is most likely inhibited? You can easily say isocitrate dehydrogenase catalyze oxidative decarboxylation, but B1 is not cofactor. Alpha ketoglutarate dehydrogenase is catalyzing oxidative decarboxylation and B1 is cofactor. B1 deficiency, this enzyme is most likely inhibited. This is important question because many times you will find incomplete statement in many books. Even textbooks have mentioned this, that B1 is cofactor for oxidative decarboxylation. But, and question is also asked, but the precise statement is B1 is cofactor for oxidative decarboxylation of alpha keto acid. Now, energetics of Krebs cycle, 3 NADH are produced. I can highlight, yes, 1 NADH is produced by isocitrate dehydrogenase. 1 NADH produced here. Okay. Second NADH produced by alpha ketoglutarate dehydrogenase. Second NADH. Third NADH by malate dehydrogenase. So 3 NADH produced. 1 FADH by succinate dehydrogenase, succinate dehydrogenase, 1 ATP or GTP produced by thiokinase. So you can say 1 acetyl coenzyme enter Krebs cycle, 1 acetyl coenzyme enter Krebs cycle to produce 1 acetyl coenzyme produce 3 NADH, 1 NADH produce 2.5 ATP. So 3 NADH will produce 3 into 2.5, 7.5 ATP. 1 FADH will produce 1.5 ATP. And 1 GTP or ATP is equivalent to 1 ATP. So net gain would be 7.5 plus 1.59 plus 1, 10 ATP. We have already discussed by mnemonic TCA, T for 10 ATPs are produced by complete oxidation of acetyl coenzyme. So 1 acetyl coenzyme enter Krebs cycle to produce 10 ATP. Now, what are the cofactor of Krebs cycle or vitamins used in Krebs cycle? Wherever oxidative decarboxylation is there, B1 is cofactor. Alpha ketoglutarate dehydrogenase, B1 is cofactor. I have already told you in link reaction, but I am repeating it again. So 3 enzymes are multi-enzyme complex. 3 enzymes. Yes, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, branch chain alpha keto acid dehydrogenase. These are multi-enzyme complex. And they are having E1, E2, E3. E1, E2, E3. And they are having 5 coenzymes. Yes, B1, B2, B3, B5, and lipoic acid. So these 5 vitamins are cofactor for Krebs cycle question, also cofactor for these 3 enzymes, and all these 3 are inhibited by arsenite. All these 3, or in fact 4, including Krebs cycle, are inhibited by arsenite. I have already discussed, but repeating it again, that active form of B3 is NAD, active form of B2 is FAD, B5 part of coenzyme A. So anywhere you find coenzyme A, you will say B5 is cofactor. Anywhere you find NAD, you will say B3 is cofactor. Anywhere you find FAD, you will say B2 is cofactor. If you find oxidative decarboxylation of alpha keto acid, B1 is cofactor. Lipoic acid, I am not explaining. But remember, in Krebs cycle, yes, I can say NAD is involved, so B3 is cofactor. FAD is involved, B2 is cofactor. Succinyl coenzyme A is there, B5 is cofactor. Oxidative decarboxylation is there, B1 is cofactor. B1 is cofactor. And lipoic acid. And all these are inhibited by arsenite. So this arsenite inhibit alpha ketoglutarate dehydrogenase. Now, first substrate of Krebs cycle is oxaloacetate, act as catalyst. Aconitase is lyase. It is not isomerase. Catalyze 2 steps. One is dehydration. Second is rehydration. I told you a crow. Previously he was thirsty, dehydrated. Then took iron pellet and put in the, yes, pitcher. And now he became hydrated. Rehydration. Dehydration, rehydration, and aconitase is Fe containing enzyme. Enzyme aconitase inhibited by fluoroacetate. This is non-competitive inhibition. Alpha ketoglutarate inhibited by arsenite. It is also non-competitive inhibitor. Succinate dehydrogenase inhibited by malonate having 3 carbon. It is competitive inhibitor. And malonate, I told you, also inhibit CPT1 of beta oxidation and complex 2 of electron transport chain. Now, what is the mechanism of this fluoroacetate? I write here. Fluoroacetate bind with coenzyme A to form fluoroacetyl coenzyme A. Actually, fluoroacetate is nothing but acetate in which fluoride is there. So it will behave just like acetate. Acetate form acetyl coenzyme A. Fluoroacetate will form fluoroacetyl coenzyme A. Acetyl coenzyme A bind with oxaloacetate. Acetyl coenzyme A bind with oxaloacetate to form citrate. Fluoroacetyl coenzyme A also bind with oxaloacetate to form fluorocitrate. And this fluorocitrate inhibit aconitase. Yes, this enzyme aconitase is inhibited by fluorocitrate. So this is the mechanism. I highlight fluoroacetate bind with coenzyme A to form fluoroacetyl coenzyme A, bind with oxaloacetate to form fluorocitrate, and this fluorocitrate inhibit aconitase. This question has been asked previously. We say that citric acid cycle is amphibolic cycle or amphibolic pathway. Catabolic, why it is catabolic? Because acetyl coenzyme ultimately converted to 2CO2 oxidized. Anabolic, 3 example, citrate is formed in citric acid cycle. This citrate come out of mitochondria, and this citrate is taking part or helping in fatty acid synthesis. So synthesis means anabolic. Okay. Alpha ketoglutarate, I told you alpha ketoglutarate, glutamate, glutamine, all are having 5 carbon. We found that alpha ketoglutarate and glutamate, the only difference is of amino group. If you join ammonia, this alpha ketoglutarate will be converted to glutamate. Glutamate can be converted to GABA. GABA is inhibitory neurotransmitter. Glutamate is excitatory neurotransmitter. You all know. So anabolic. Now You all are aware that heme synthesis first step occur in mitochondria and succinyl coenzyme A bind with glycine to form delta amino levulinic acid. By delta amino levulinic acid synthase, heme synthesis ultimately lead to heme synthesis. So heme synthesis anabolic. So you can say this is amphibolic cycle, both catabolic and anabolic. Three example I have given. All these are questions. Anaplerotic reaction. Replenish intermediate means form the intermediate, fill the gap means pyruvate is converted to oxaloacetate. This is intermediate of Krebs cycle or starting point of Krebs cycle. Means pyruvate is having 3 carbon, oxaloacetate is having 4 carbon. A 3 carbon compound is converted to 4 carbon compound means 1 carbon must be added. So CO2 is added. Joining of carbon dioxide is called carboxylation, and that's why the name of enzyme is pyruvate carboxylase. And wherever you find carboxylase, yes, carboxylase mnemonic is ABC. Carboxylase use ATP, A for ATP. Yes, use biotin as cofactor. Biotin as cofactor. C for carbon dioxide is joined or bicarbonate ion is used. So ABC. So first anaplerotic reaction is pyruvate to oxaloacetate by pyruvate carboxylase means pyruvate produce oxaloacetate, that is the part of Krebs cycle. Now second anaplerotic reaction, phenylalanine and tyrosine are converted to fumarate. A question is asked in a different way that fumarate of Krebs cycle is produced from which amino acid? Simple mnemonic, fume. Fume comes from tire soap. If you have tyrosine, I remember tyrosine as tire, the soap of tire. So bad fume you will find tyrosine. Phenyl bottle, if you open the phenyl bottle, so bad fume, phenylalanine. So phenylalanine and tyrosine will produce fumarate. Next, valine isoleucine methionine, Vim bar, produce succinyl coenzyme A. If you want your girlfriend or boyfriend to wash clothes nicely and successful washing, you will give him, yes, Vim bar, advertisement of Vim bar, successful washing के लिए use Vim bar to give to your boyfriend. Yes, so valine isoleucine methionine. Now, I told you glutamine, glutamate, alpha ketoglutarate, all are having 5 carbon. The only difference is ammonia. If ammonia is removed, glutamine is converted to glutamate. If ammonia is removed, glutamate is converted to alpha ketoglutarate. So these are anaplerotic reaction. Questions mostly asked on pyruvate to oxaloacetate by pyruvate carboxylase. Now regulation of Krebs cycle. Krebs cycle is very vital. I have already told you that carbohydrate, lipid, protein, ultimately the final oxidative pathway is Krebs cycle. If Krebs cycle is defective, the cell will not able to survive. It is lethal. That's why nature said that this is so important for the energy production. We will not give control under hormonal regulation. Hormonal regulation is just like the central government. We will say no, no, no. We will regulate this Krebs cycle by allosteric regulation. Local government. Allosteric means local government. Means local government will decide what is the requirement of the local population. Means if cell require energy, TCA cycle will run. If cell don't require energy, TCA cycle will stop. So Krebs cycle has no hormonal regulation. Question. Regulation depend on energy status of the cell. If NADH produce how many ATP? 2.5 ATP. ATP increases means energy is sufficient in the cell. So cell will say stop cycle. Yes, decrease TCA cycle. If NADH ATP decreases, energy decreases, then yes, you must activate TCA cycle to produce ATP energy for the cell. Decrease NADH ATP, increase TCA cycle. I have already told you regulatory enzyme. Three regulatory enzymes are there: citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. Three regulatory enzymes are there. Now, complete energetics of glucose. Glucose is converted to two molecules of pyruvate. Aerobic glycolysis, net gain was 7 ATP. I told you aerobic, mnemonic. Yes, aerobic, 7 letters are there, 7 ATP. Now, two molecules of pyruvate enter the mitochondria with the help of H ion. HN import. Pyruvate is converted to acetyl coenzyme A by pyruvate dehydrogenase. In this, two NADH are produced, and you know, as two molecules are there, so two NADH are produced, and you know, one NADH produce 2.5 ATP, so two NADH will produce 2.5 into 2 is equal to 5 ATP. So till now, 7 ATP by aerobic glycolysis, 5 ATP by pyruvate dehydrogenase link reaction. Now, two acetyl coenzyme are produced, and I told you calculated also one acetyl enter Krebs cycle or TCA cycle T4 TCA cycle T4 10 ATP are produced, but two acetyl coenzyme are there, so one acetyl 10 ATP, two acetyl 10 into 2 20 ATP, so 20 ATP plus 5 ATP plus 7 ATP, that is 20 plus 5 25 plus 7 32 ATPs. So complete oxidation of glucose produce or net gain, better to say net gain by complete oxidation of glucose 32 ATP. Thank you.